session is going to be a repetition of what we started last week. I'll refresh your memory a bit, and then I will introduce the two mouse models that we are still missing.
Professor, are the slides posted for this week?
The slides were posted, yes. I did it earlier, so...
Okay, I see it now. Thank you.
They were there. I was just modifying some of them. So. Okay. So could you please confirm that you're seeing my slide? Okay, great.
Yeah, we can see it.
Okay, thank you. Recording is there. Okay, so as for today, as I said, we are gonna finish up with the transgenic models. I must tell you that I'm explaining to you the general concept of transgenic animals. So we started discussing how to generate a transgenic non-specific animal where you just insert the DNA, inject the DNA, and it will be inserted in multiple copies in different locations on the chromosomes or sites that you cannot control. And then we moved into generating a knockout animal, which means we are knocking out or deleting a gene. And this is somehow also not controlled because if you remember, we mentioned that in conditions where you delete certain genes, it could be lethal, and therefore you cannot study that mouse model. So for that, we get into some modifications in order to generate conditional knockout animals. And in order to be able to generate conditional knockout animals, more tools were engineered, particularly those knock-in animals. And as the name indicates, knock-in is to introduce a gene or a DNA sequence into a specific target, into a specific site of a gene. So we will be describing this by the end of the lecture. But in order to use these knock-in animals, we have to set the ground by producing inducible and tissue-specific models. So it we need kind of go back in a reverse step. We describe first what is an inducible and a tissue-specific model. One part of it would be the usage of the knock-in animal. So just bear with me because okay. So we agreed that transgenic is accepted to be an overexpressor of a gene and knockout is a loss of a gene. And I gave you an example, leptin knockout. And you can see the difference between a wild type animal and a leptin knockout animal. Now, the issue here is that when we generated this leptin knockout animal, as I said, we most probably deleted some exons from the leptin gene, and this animal is created. However, we can't anymore control the knockout. In other words, if I want to knock out the gene at a specific age of this animal, I can't do it because it's a general body knockout. When the embryo is generated, after fertilization, we have the knockout and that's it. So that's why we start to think about conditional knockouts. And I want to remind you that in transgenesis, the injection of DNA that will be inserted unspecifically, it would be at the just after the oocyte fertilization. Most of the knock-ins, knockouts, and tissue-specific inducible mice, we use the second method, which we described using the embryonic stem cells. So most of what I will describe today is going to be using the embryonic stem cells. Just to remind you, so for those who were not in the lecture last time, or probably you didn't review the material, in order to generate embryonic stem cells, obviously we have to mate. So there's one step that is not on the slide. We have to mate wild type animals, male and female, and then we will have an embryo, and from that embryo we will isolate the embryonic stem cells. We will target those cells with the vector. So after transfecting those cells with the vector or electroporating the vector, we will get some of these cells transfected and some not. And there will be the homologous recombination step that we will test. And after that, we will need to put those positive cells after the transfection and after this selection, we will introduce those back into a mouse that is pregnant. And this mouse will give an embryo. So we took those cells, we put them in a pregnant mouse, and this mouse will generate an embryo. And that embryo will carry what we say chimera. One part of the embryonic stem cells come from the engineered embryonic stem cells that we had working on the lab, and another part of the embryonic stem cell come from the actual embryo in the mouse, and therefore we have a mix of the two, and that's the chimera. And after that, we have to breed these animals couple of generations in order to get the full body knockout. So first, we have to take embryonic stem cells, engineer them, and interrupt the gene we want. And second, we have to reintroduce those back to mouse, and then breed that mouse and probably breed it into 2 generations to get these steps. Okay, so if anybody is not able to understand these 2 steps, please stop me now and let me explain again, because this is very important for the next few steps. Any questions, any concerns, any of these steps are not clear? Okay. You don't make the embryonic stem cells. So how one of your colleagues is asking how to make embryonic stem cells. So you need to the generation of the embryonic stem cells, as the name indicate, it's embryonic stem cell. So this means you have to take one mouse and another mouse, you breed them together, and they will egg and sperm, making a zygote, which is a fertilized egg, and then it will grow to an embryo. At certain stage of the embryo, if you recall, I will go back in a few slides. So that's what happened here. When we mate male and female mice, the fertilization step, and then the fertilized oocyte will become 2, 4, 8, 16 morula, and then that's the embryonic stage where we get the embryonic stem cells from, at this level here, when we have blastocysts. So they are collected from, yes, they are collected from female, because embryos are growing in females. Okay, then, So that's it. We breed the animals, we keep the embryos growing until certain time, and then we take the embryonic stem cells. And at this point, now we have embryonic stem cells in culture, and we have on the other side the vector that we cloned. And I said before, there's a lot of studies you have to do in order to know which gene you are gonna work on, which exon you want to delete, and all those information before starting such a project. It's something really big what we're doing here. Now, the next step is that I engineered those and I modified, deleted whatever gene I want. So now I have embryonic stem cells that are engineered. I will repeat again the breeding, but now in a different mouse line. So if this is brown, this will be white. If this is black, it will be brown, just to get the difference between the two animals. Now I'm breeding again these two mice. They will generate an embryo again. And I will let the embryo grow until a certain point where it's the plasticist again. And at this point, I will get those engineered embryonic stem cells that were selected into the embryo. And that's how I create the chimera of mice. Pretty much, then there's breeding and there's the genotyping process. We went through this in the last lecture. So when we are generating the vector, the gene in question, we interrupt the gene by neomycin cassette. So this neomycin will be replacing part of that gene. And that's why you see the green part is interrupted by a red, which is the neomycin. And we add on the extremity of that gene, thymidine kinase gene for selection. Three outcomes of the embryonic homologous recombination might result. The first one would be there will be no recombination. Recall that this is the genomic DNA and this is the vector. So here there's no recombination because the DNA didn't change at all. In this condition, the cells, they don't have neomycin resistance, they will die. In conditions where the recombination took place perfectly, it will recombine using the homologous arms, the homology arms around the gene, and therefore it will lose the selection with the thymidine kinase. With this type of selection, if I place the cells with thymidine kinase drug, the cells will not react to that and they will survive because they don't have the thymidine kinase gene. And this is the recombination we are looking for. What happened is the recombination replaced the part of the green gene on the genome with the part that we want to modify. Great, those cells are resistant for neomycin and they will not be sensitive for the thymidine kinase drug. Now, in the third situation where we have unspecific or non-specific recombination, we will carry over the thymidine kinase gene, and if we add the drug for the thymidine kinase, it will kill the cells. Therefore, even though the recombination took place, it's non-specific, but it will be eliminated too. And therefore, with this strategy, we end up eliminating those cells, and we'll have only those red cells selected. Those are the ones you inject in the second step here into the mouse embryo. And those are the ones generating the chimera. And after that, as we explained before, you will breed the mice a couple of times. First, you have a heterozygote, meaning one copy of the gene is replaced and the other one is a wild type. This is in the second generation. And if you breed those together, you have a possibility where you get a mouse that carries the two homozygote knockout gene. So we kind of stopped here last time. We discussed that you have to genotype the mice, meaning you have to examine if you have copies of that DNA changed, and you have to phenotype the mice, meaning look what is the effect of that knockout, meaning if you are looking for a if you're thinking that this gene is gonna induce cancer, you will take this mouse and you start testing if it's having tumors or not. Sometimes, as I said, the knockouts are lethal, and that's a limitation. We can't do much here. Sometimes the knockout doesn't give us the result we expected because maybe there is splicing that's happening. We remove exon number 2, but then the protein have another transcript with exon 1 and exon 4, and we still have the function. And I gave you an example here of the p53 knockout, which is not embryonic lethal, which is great. But since we knock out the p53 that is a tumor suppressor, we lose the suppression properties, and therefore this knockout mouse develop a lot of tumors. And this is an example of how we look at these knockouts in publications and how we present it, in fact. So the example here is showing you that we want to target this gene and remove exon number 2. So this is a genomic locus. Obviously, I know the sequence. So I know that this is a HND3 restriction enzyme, EcoR1, another HND3, another EcoR1. The homologous arms are between the HND3 and the EcoR1 on this 5' end. And again, between the HND3 and EcoR1 on this 3' end. So that's how so between the two arms, the homologous recombination takes place. And in order to eliminate exon number 2 and replace it by the neomycin cassette, we generate this vector where we insert the neo on that vector, retaining this homologous arm and this homologous arm, or arm of homology. Now, when the recombination takes place, neomycin cassette is going to replace exon number 2. So in order to know that the process has taken place, and in order to genotype the mice, in other words, to make sure that the neomycin cassette replaced exon number 2, I have to do two things. First, to generate a primer that is specific to the neomycin cassette on one end, and to the homology arm on the other end. And therefore, if there is a PCR product that comes out from these two primers, 1 and 3, it will tell me that this mouse carried the neomycin cassette, and therefore there's a great possibility that exon number 2 is removed. On the other side, I will have also to generate a primer specific to exon number 2, and you can see the same primer number 3 is common between the two conditions. Okay, now what happens is sometimes, like when you are genotyping, obviously you can get three possibilities. One possibility is that you have one copy of the neomycin cassette and one copy of exon number 2, and that's the heterozygote you get in the second generation. You can have the possibility where two copies were replaced by the neomycin cassette, and that's what you get in the third generation. So if you look back here, this is the heterozygote, so you have one copy and one control copy, which is exon number 2 is still here and neomycin is there. And you can get this situation, we have both having the neomycin cassette. So having this PCR strategy, then you would be easily able to genotype the mouse and know if it's a homozygote or a heterozygote for the knockout. I see some questions. I will answer those just when I finish this. So the other thing is, if you want to utilize the southern blotting as a genotyping strategy, we can see clearly here that we lost one echo R1 site. And recall, when you do the Southern blotting, you need to digest the genome with a restriction enzyme. So you will isolate the DNA, digest with a restriction enzyme, which is an echo R1 in this situation, and then use a probe to detect that gene. The probe should be in this area here. Now, if I'm like let's say the probe is between the HEND3 and echo R1. So that's the probe. So in conditions where you have, so it has to be a little bit further than this one here, like somewhere here. So in conditions where you have the exon number 2 not replaced, then you will get two products. And the product here is the echo one, this is small one, and that large one. Okay. Now, in conditions where you deleted exon number 2, you lose that, and therefore you will have only a chance to get that large echo R1. So this is what you see in the Southern blot here. In the wild types, you will get this small product. In the heterozygote, you get one small for the wild type and one large fragment for the knockout. So this is if we use Southern blotting for genotyping. Now, if we use the PCR, the first product comes between primer 1 and 3, and the second product comes from primer 2 and 3. So in the wild type, you have exon number 2, and you made your strategy that the PCR product between exon 2 and exon primer 2 and 3, it will be less than 400 base pairs, which is this. So in conditions where you have 2 copies of exon 2, you will have that. And in conditions where you have 2 copies of the neomycin cassette, you will get a little bit bigger PCR product between 1 and 3, and that's going to be here. And if you have 1 copy and 1 copy, which is the heterozygote, you will get 1 copy of each. So one PCR product comes from the 1,3, which is the upper one, and then one PCR product comes from this one here. Okay. Later on, we will check the protein. Forget about this at this point. Okay, I'll check what were your questions. So the homozygotes are only observed in the second generation and up. Sometimes you need so your colleague is asking, when do we see the homozygotes of the mice for the knockout? Sometimes in the second generation, sometimes you are not lucky. You still have to breed one more generation, but at least in the third generation, you start to see, not in the second generation, in the third generation. So if you go back to that slide, which is here. So this is the first generation. This is the first pop you get from the embryo after. So that's the mouse where you put the embryonic stem cell. That's the first generation animal, which is a chimera. If you breed this to another mouse, that's the second generation where you get heterozygotes. And then you have to breed those like this to that, probably to get you one homozygote. So in the third generation, you start getting the knockouts. Okay, so now we have transgenic animals versus knockout animals. Remember the differences and recall that in the transgenic animals, when we generate those, we are trying to carry or introduce certain genes that started to be in a non-specific manner by simply injecting DNA to fertilized egg, to the pronuclei of the male. We inject the DNA in a linear fashion, and the insertion of that DNA into the genome is random. While in the knockouts, it's more specific to a certain gene. So it's really corresponding to one gene that is specific. It's probably one exon or two exons of that gene that will be deleted. And it's widely accepted that knockouts are more like the knockout process is more laborious because you have 2 times to isolate embryonic stem cells, and you have to make sure your replacement vector has been generated and validated. So the replacement vector is transfected into the embryonic stem cells. So we are not introducing linear DNA, we are introducing a vector, and the recombination takes place for only one gene, and it requires multiple generations to validate these knockouts. So these techniques can be used in other organisms. We are just describing in the mouse, but bear in mind it can be done in other organisms. And there is another strategy to generate knockout animals. This would be by using the CRISPR. I will evaluate if I will explain this to you in the next lecture or not. I just wanted to keep it flowing with the sessions. So probably in the next session, I will take 10-15 minutes to explain the CRISPR knockouts. Now, one of the applications that came after this long time of practicing the transgenic animal generation is the in vitro fertilization. Without all this laboratory work on animals, we would not be able to perform the in vitro fertilization that nowadays we utilize for generating embryos and implanting them into women. So exact same procedure that we are doing in the mice, we learn a lot from how the half-life of the sperm, the isolation of that sperm, and the ability to inject things to that sperm. So all those techniques and experiments gave us a lot of information of how to manipulate an egg. And therefore, this is now in practice. You know, there's a lot of in vitro fertilization in clinics where people are not able to get like women not able to get pregnant would go to this type of procedure for multiple reasons. Could be due to the amount of the sperm or the quality of the sperm, or probably the conditions where the eggs are being generated. So for that, first we have to, and recall I don't want to make a comparison between a human and an animal, but recall that most of these terms we just described before. So when we said prepare a pseudo pregnant female. So for example, it's not in this slide, probably in the previous one. So recall when we said you will take the nuclei, take the eggs just after fertilization, where we have the sperm nuclei, just the pronuclei just inserted into the egg. So that's the egg nuclei, and that's the sperm pronuclei. Nucleus, sorry, I'm saying in plural. We know that the sperm pronucleus is bigger in size, and that's where we inject the DNA for the transgenesis. Now, we're doing mostly that, letting the cells grow, and then we will insert them to a pseudo-pregnant mouse. In other words, we will inject these mice with certain hormones to get prepared for pregnancy. So this is one thing. Now, when we are discussing about the in vitro fertilization, we will introduce the stimulation for ovary in order to generate eggs. So the woman who wants to go for the in vitro fertilization has to go to this stimulation. And therefore, we will have a specific amount of eggs that will be generated and ready to be isolated. So you know that females can have maximum 2 eggs, right? And we will take those eggs and then we will fertilize them in vitro, the similar way that we discussed. But now, instead of introducing the DNA into the eggs, we are introducing the whole sperm into the egg. And then we will leave those grow for a few, I don't recall what exactly, how many days they keep this. And then again, you will stimulate the ovary of the female to be prepared to carry the embryo, and then you will introduce those embryos back. And recall, many times when we are describing those processes, you have to know that we are not doing only one egg and one sperm. We are doing more than one egg and more than one sperm. So that's why one of the problems that might happen is that the risk of having multiple pregnancies, which is you have triplets or more. And obviously there might be ectopic pregnancy where the implementation of the embryo could be in the fallopian tube, not in the uterus. So that's also problematic. And of course, there are many other side effects. There is a video for you here. And that being said, again, I don't want to compare what we do on mice similar to human, but this is one of the fruits, if I want to say, of the whole procedures of learning all these techniques and genetic engineering on mice, that now we can apply something like in vitro fertilization in human. Okay, which brings us to the second part of today's lecture, continuing on the generation of tissue-specific and conditional knockouts. So if you understood what I have said about the general knockout, it will be easier now because it's some more specific details will be added to generate conditional knockouts. So as I said, one of the major limitations of the knockout, whole body knockout animals is probably its embryonic lethality. And the other thing is that we can't control when we want to knock out this animal. Imagine, for example, you are studying an aging-related gene. So you want to knock out this gene with age only, not like in the embryonic stage. The conditional knockout will allow you to do that. So experimentally, there are multiple systems that are described. I'm not going to go into details onto all the systems that are being used to generate conditional knockout animals. We will focus on the Crelox system. Basically, instead of, let's say this is exon number 2 that we were discussing multiple times on the slides. If you generate a whole body knockout or a knockout, you delete that you delete that in the embryonic stage. Now the Crelox system allows you, instead of deleting the exon, you can introduce LOXP sites around that exon. And then whenever you want to delete the exon conditionally, whenever, like, let's say at the age of 20 weeks of mouse age, then you can use the CRE, which is a recombinase that clips that exon and deletes it when the mouse is adult. So this is a very elegant system in order to delete genes. The components are the CRE recombinase, which is a tyrosine recombinase. It's derived from bacteriophages. And the CRE recombinase recognizes very, very specific sites to perform the recombination process. Now, these sites are called LOXP sites. And in order for the CRE to excise that exon, there must be 2 LOXP sites around it. So the CRE has to recognize these 2 LOXP sites and then perform the excision. Now the LOXP sites, I will describe them in the next slide. These are very short 34 base pair DNA sequences. So we know the sequence of those LOXP sites. There's another system which we call lipase and FRT, which is exact same thing. The only difference is that the lipase is instead of being from the bacteriophage, it comes from Saccharomyces cerevisiae, which is a yeast. So there's a slide coming to compare the two systems further. So now the Cre recombinase recognizes 2 repeated 34 base pairs. Okay. And this is the sequence for the LOXP site. So the end product is that if I have 2 LOXP sites and I then express the protein Cre, which is the Cre recombinase, then I will remove that exon or whatever sequence in between. Now, in order to excise the sequence in vivo, this is more complicated than just the scheme that I'm generating here. So first, you have to generate a mouse that carries the 2 LOXP sites around the sequence that I want to delete. And then you have to generate another mouse that expresses the Cre. And then you cross these together. And then the Cre that is generated is going to remove that gene, and you will end up deleting that gene. So you have to generate 2 different mouse lines, cross them together, and then look at the results. Okay? And please stop me if you are lost. So both LOXP site, flank gene, and Cre recombinase has to be crossed within the same mouse in order to perform this conditional knockout. Now, I'm repeating this because many times I'm saying things and some of you saying that most of the things I talk, they are not written. So I wrote it down in the slide. In order to generate a tissue-specific model or mouse model, we require to generate 2 mouse lines. The first mouse line, it should carry a LOXP flank gene, whatever that gene, and it should be in 2 alleles of that gene. So if you go back here, if you have only one that is a flanked and the other one is a wild type, then you are having a heterozygote animal. So 50% of the gene will still function. So in order to delete totally that gene, you first have to express the Cre, and also you have to have a homozygote conditional gene. And then the other mouse should express the Cre recombinase, and this is generated, both are generated by the embryonic stem cell injection method that we described for the general knockouts. Now, for the Cre, I just want to introduce here the notion that you can specify which cells can express a Cre and which cells do not express the Cre. So if I go back a step, it's saying expression driver or a promoter, right? And the examples I put for you here, the albumin promoter is active only in the liver and the hepatocytes. Therefore, if you generate a mouse with albumin inducing albumin promoter and the Cre is under control of that promoter, then it will work only in the liver. Similarly, PAX7 promoter only works in skeletal muscle stem cells. Alpha myosin heavy chain promoter induces Cre induction only in the heart cells, in the cardiomyocytes. Insulin promoter induces expression only in the beta cells in the pancreas. So you already start having, from these couple of slides, 2 levels of control. The first level of control is that instead of deleting that gene by the general knockout procedure, now you have ability to decide when you can delete that gene. So you have the gene there till the time that the Cre is generated. And the second thing is, by choosing which promoter to induce the Cre expression, you can choose which cell types you want to delete that gene. Okay? Now, just a comparison between the CRELOX-P system and the FRT system. So, and remember that we are studying genetic engineering and recombinant DNA technology and transgenics, but we are not studying everything. So I'm just presenting for you here some of the other functional aspects of the LOXP site. The LOXP sites and the Cre recombinase, they are exact same function as the FRT sites and the flipase. So you can see that the sequences are known, there's a spacer, and then there's an inverted repeat. Wherever the Cre recombinase recognizes the inverted repeat on this side, the LOXP site across a target gene, so in between these two, it's gonna go and perform the recombination process. But now the recombination process doesn't mean all the time it's deletion. In nature, there are multiple outcomes. One of them is deletion if we perform adding the LOXP sites in parallel to each other, like one after the other. But if we inverted the LOXP sites, we have simply an inversion. And if we have only one LOXP site, we will get a translocation. We are not discussing those two. I'm just telling you that it's not always the LOXP site and the Cre recombinase gets into deletion. This is how we use it, because we know that if we put the two LOXP sites, one downstream the other, then the process will be deletion, because with this we have tested in the lab and we know it. But if the sequences were inverted, then will be inversion, and there's no deletion, and there's no, if there's only one copy, there will be translocation. We are concerned in this lecture, in this session, or transgenic animals, just using this, because nowadays all transgenic animals use this. That being said, you have to be super careful if you are generating your plasmid, and be careful that these two LOXP sites has to be one downstream the other in the same order. Okay, I see a question. Okay. Does the Cre recombinase line also have to be homozygote? No. The Cre line doesn't have to be homozygote because you are overexpressing the Cre. The Cre will be a protein that you are generating at. Now, many times we try to have it heterozygote, not homozygote. I am sorry, I am slightly confused. How do you control when Cre will cut out the exon between the LOX two sites? So your colleague is asking, how do you control? You control only, like what I showed you so far, you control by which cell type. Because if you are expressing the Cre under the control of pancreas, specific promoter, which is insulin, the expression will happen only on those cells. But how, you will see in the next few slides. Okay, now, the FRT flipase is exact same thing, and it does the same function. The only difference is, as I said, the flipase come from the Saccharomyces cerevisiae, not from the bacteriophage. And the flip sites are exactly like the LOXP site in terms of function. They are difference in the sequences. Okay. Now you will see that we will merge both LOXP, Cre, and FLP for FRT systems together when we are generated the inducible knockout animals. So let me go back and refresh your memory how we generated the general knockout replacement vector. The general knockout replacement vector has homology arms toward the gene of interest. So if we are talking about exon number 2 on whatever gene, then we have to use those homology arms on exon 1 and 3. and it's represented for you here. So this is the genomic locus that we want to delete exon number 2. We utilized exon 1 and exon 3 as homology arms for the recombination to take place. And we introduced, instead of exon 2, the neomycin cassette. And at the extremity of the gene, we added the thymidine kinase selective gene. Okay. Now, when the recombination took place, neo replaced the exon 2. And the correct homologous recombination should not carry this guy. Otherwise, the embryonic cell will die. And the end product would be that exon 2 is only replaced with neomycin cassette. Now, in the previous lecture, I mentioned, but it was not written on the slide, that the thymidine kinase gene can be, there are other alternatives for that. One of the alternatives is diphtheria toxin A. And the diphtheria toxin A gene, if you add the diphtheria toxin for those cells that have wrong recombination, like in the second condition we described, then those cells will die. So if you see in some of the vector thymidine kinase gene or diphtheria toxin, they both perform the same function, which is killing the cells that have unspecific recombination. Okay, now how does it look like in the conditional knockout? So remember, this is the step that we are describing, where the recombination will take place around the homology arm of the site of exon number 2. Now, there is some slight differences that we added to this replacement vector. So this, again, we have the homology arm upstream, the 5' end, and we have a homology arm downstream. So therefore, the recombination will take place around exon number 2. If there's anything changed, it will be this. But now, instead of having only neomycin cassette, we have the neomycin cassette, and we have exon number 2. But around exon number 2, we added the LOXP sites. Okay? And around the neomycin cassette, in case in the future we think to remove it, we have the FRT. Okay, so we have the two systems here. The FRT flippase in order, if we want to remove the neomycin cassette in the future, because we are introducing a change that will happen for life in that mouse. And the time that we want to remove the exon number 2, we can use Cre recombinase because it will recognize the LOXP site. So if the right recombination takes place, see that here you have the DTA as a selection, not the thymidine kinase. The correct recombination requires that this doesn't carry into the genome, and only this site is flipped, and exon number 2 is excised, and the neomycin cassette exon 2 is introduced. So now what happens? At this point, we don't have issues in terms of the sequence. We didn't interrupt the sequence, because at this point, we have the exon number 2 still there. Okay, so in the next step, what we need to do is simply excise the neomycin cassette. So that would be one level of selection that we can do. So we remove the neomycin cassette, and we end up with the product that is exactly the same with this guy, with the difference is that we having 2 LOXP sites around. Okay, I just put a note for you here. I don't want you to know this for your exam. Sometimes you read PGK neo. It's just a modification of the neo cassette. Okay, just in case you are asking this question, what's the difference between the PGK it's written for you here, but I don't want you to know what's the difference between PGK and regular nail. Okay, so the end product of this conditional knockout targeting vector is that simply in 2 steps, I introduced first a gene, the exon with a LOXP sites, and then I also have the FRT around that neomycin cassette. When I remove the neomycin cassette by flippase, I will end up with a product where I have exon 2 conditionally can be removed whenever I express a CRE. Anytime I express a CRE, I can remove exon number 1 in this condition. Okay, any questions here? Okay, no questions. Great. Now, so this is the first level. I generated a conditional exon number 1. And the second level of control is the expression of the CRE. And I kind of mentioned a little bit in the previous slide that the condition of the CRE where it's generated, it depends on which cell type. So the promoter region defines where in the tissue or in the cells, in which cells or tissues the CRE can be expressed. The CRE can be expressed globally, for example. Remember the mouse that we discussed with the ubiquitin C. The GFP was expressed everywhere. So we can, if we want to generate a mouse with a CRE in all the cell types, we can use ubiquitin promoter. But if we want to make a tissue-specific or a cell-specific CRE expression, so we have to find that tissue-specific promoter. What is it? So if you are working on the heart, there's only one alpha-myosin heavy chain. I just wrote it in the previous slide. If you're working on beta cell of the pancreas, insulin promoter. If you're working on stem cells in the muscle, it's PAX7. So we know from many, many years of research, certain tissue-specific promoters. So we take that promoter and we clone the CRE downstream of that promoter. Okay? And what will happen is whenever we generate that CRE in this cell type, let's say the example here is beta cells of the pancreas. If this is beta cells of the pancreas and this is insulin promoter, then the CRE will be only generated in the insulin generating cells or beta cells. And if you are having another gene where it's flanked by LOXP site, you can delete that gene only on beta cells of the pancreas. Now, if you are generating a promoter that is not tissue-specific, it would be this condition here, like ubiquitin. It will be expressed everywhere. What happens in the non-target cells, like if this is insulin promoter and that's the CRE under the control of insulin promoter, in other cell types, like in the skin, in the heart, in the in the kidney, there is no insulin generated, and therefore the CRE is not generated, and therefore there will be no deletion of that gene because the CRE is not there to go and excise the LOXP sites. Okay, so that's how the mechanism, it works in a tissue-specific manner. It's written for you more detailed here. Now, the second level of control of a CRE expression, it's the inducibility. So not only to generate a CRE, but to generate a CRE that is not functional unless we add another control on top of it. And that's going to be the Merck-Remer or the CRE ERT2. So in the inducible CREs, again, you can still use a tissue-specific promoter. Like you can still produce this CRE only in the pancreas, in the beta cells of the pancreas, because you are using the insulin promoter. But the problem is that you are not the problem, but the modification is that it's not only a CRE, it's a Merck-Remer or a CRE ERT2. And I will describe what are those in the next slide. How does it work? Now, when you generate the CRE, what happened is it binds, like here it's describing for you the CRE ERT2. okay? So when you are generating the accree, it will be bind by, it will be fused with a modified estrogen receptor, which is a mutated form of the estrogen receptor. And whenever you are generating the accree, it will be retained in the cytoplasm in the absence of the exogenous ligand. In this situation, that's a tamoxifen. Only in conditions when you add the tamoxifen, then it can go to the nucleus and perform the cutting. So the modification is, instead of generating straight active accree, you generate a modified accree that is bound with a modified estrogen receptor or mutant estrogen receptor. And then you have to add tamoxifen, which replaces this HSP90 protein, so that the accree becomes active. When there is no tamoxifen, there is no excision and there's no deletion. The gene stays intact. Whenever you add tamoxifen, then tamoxifen will replace the HSP90, and then the CRE, ER, tamoxifen together go to the nucleus and excise the gene, and now you have a knockout. So now you have a tissue-specific promoter, which means in this condition it's insulin, beta cells. Only insulin and beta cells produce the CRE. If you are using it here, only the insulin, beta cells producing the CRE, ER, HSP90. And only in conditions where you add the tamoxifen, this CRE will be active. So you will need to have the tamoxifen on board in order to excise that gene. And this is the most elegant system and the most widely used these days, because if you want to delete the insulin promoter on the age of 100 days, all you need to do is simply generate this CRE, cross it with the gene of interest, and on day 100, you just inject the mice with tamoxifen. And then on the day 100, you start having the deletion of that gene. Okay? Questions? All clear? No questions. All clear. Amazing. Okay. So if I summarize the process, the conditional knockout process is 2 steps. The step number 1 is to know which gene you want to flank, and you know which exon you want to flank, and then you generate a homozygote flanked with the LOXP site. So you have to know that the process I described for you for the general knockout is exactly the same for generating the LOXP site. So you have to generate the homozygote by multiple generation, at least generation 3 or 4, where you have both copies of the gene, both alleles of the gene are flanked with the LOXP site. So this is a homozygote LOXP site for the gene of question, whatever gene that you are studying. And then you would need a mouse that expressed the CRE. In this situation, it's albumin CRE. It's not inducible CRE. It's just the general CRE, but it's under the control of albumin, so it will be expressed in the liver. So it's a liver-specific CRE transgene. Bear in mind that in this mouse, when you generated, again, the same procedure, after a couple of generations, you will have the albumin CRE in that knockout mouse. Well, this is transgenic mouse, sorry. And you have to cross these together now. You take number 1 and mouse number 2. In the first generation, you might have 50% of the animals having albumin CRE. And since you have 100% wild type X here and 100% floxed here, you have a 50% possibility in the first generation. You will have 1 copy from here and 1 copy from there. And in this situation here, we have one copy. Probably you will have it or not. So you have 50% chance. So you will get at this stage, mouse number 3, where you have the LOXP system with the Cree. But so if you take this mouse and start studying it, only 50% of the mouse gene is gene X is deleted because you still have a wild type gene. So in order to get 100% deletion of that gene, you need to take number 3 and cross back with mouse number 1. So if you take number 3, I just presented again here. So this is number 1, which is homozygote for the LOXP sites. And now you will have 50% chance you get one copy from this and the same copy from this mouse. And therefore you have a double homozygote flanked gene X. Also, there's a chance that this doesn't have the Cree and this have a Cree, so there's a 50% chance that you get that Cree. This mouse now expressing Cree under the control of albumin only in the liver cells, and the gene X is going to be deleted in this mouse. Okay? Now, the issue here is that this is not an inducible Cree. This is just a tissue-specific Cree. So it's a tissue-specific, but it's not I would say controlled. In other words, maybe in the embryonic conditions, the liver cells of that mouse might have an issue because the Cree is not controlled. And that's why it's better to use the Merck or the Cree ERT2. But the good thing is that in this mouse, only the liver cells will have deletion of the gene X, not any other cells. Only liver cells because it's under the albumin promoter, because the Cree will be generated only in the liver cells. Okay, so this is a good level of control. So there is a question. Okay, just to clarify, for generating the LOXP site mouse, you will follow the same steps of the regular knockout using injection into embryonic stem cell method, and you will have to wait few generations. Exactly. So Anna is asking how we are reaching the step to generate this animal. Yes, what you wrote is 100% correct, Anna. So, and I will clarify it one more time for you guys. So in other words, to generate this animal and to generate that animal where you have an albumin Cree and this, so to be specific, to generate this animal here, you need to do all the steps that we described here. So you have your target vector, but the target vector is different. The only difference is the target vector. And then you will first mate animals, take embryonic cells, take that LOXP targeting vector, change your embryonic stem cells, select them, choose the right recombination, kill the one that has wrong recombination, kill the one that don't have recombination, then again breed 2 animals, grow an embryo, and at this stage you add your LOXP modified embryonic stem cells. And then you have to wait to get the chimera and again go to generations of breeding. So you put your the cells of LOXP site modified gene into an embryo mouse that has been pregnant, and then you will get the first generation chimera, breed again, you get homozygotes, heterozygote, breed again, you get the heterozygote. So all that you did in order to do that mouse that I'm showing. The only exception is that you're using this type of targeting vector and not this type of targeting vector. And then you will have this mouse. Okay? On the other side, you are generating an albumin tree, which is the same procedure. And now you have one mouse line and one mouse line, and now you're crossing these together. You get a heterozygote, and then you continue as I described to get the albumin tree with the homozygote. And at this point, you can start studying the animal. So this is almost 2 years of work. Excuse me. Okay, so now one little modification we will add, which is the knock-in animals. Everything I just described in this mouse here, this guy, number one, we can perform it, but this stage, instead of just adding LOX-P sites, we can add a CRE or a specific gene, whatever that gene is. Okay? And since we know that the mercurimer is controlled by tamoxifen, one of the methods that we can control tissue specificity knockout is by using first a promoter that is specific to a certain tissue. Let's take an example, the alpha myosin heavy chain, which is listed here. The alpha myosin heavy chain promoter induces a CRE expression only in the heart cells and the cardiomyocytes. So we can take that as an example. And if we want to express the mercurimer only in the cardiomyocytes, we can excise the mercurimer by the same method that we described many times now with embryonic stem cells into an exon under the control of the alpha myosin heavy chain. And what happens is whenever the alpha myosin heavy chain is turned on, the mercurimer will be turned on. Now, in this particular example, troponin or titin, I'm just confused between the two. I can make sure which one is it. We knocked in the mercurimer under the control of this promoter. So whenever this TNN promoter is active, it will produce mercurimer. And most of the time, whenever we introduce a knock-in animal or a gene, it has to be inserted to the first ATG of the first exon. So this is a super specific insertion. In other words, not always the exon 1, sometimes probably there's an isoform of a protein that starts with exon 2. It should be the first ATG of that gene. Okay? And the way it works is going to be explained in the next slide here. So now imagine you are generating a heart-specific knockout gene that is inducible. So it's an inducible and conditional at the same time, and it's tissue-specific, 3 levels. Level number 1, it is cardiac-specific. Why? Because it's a cardiac-specific promoter. Cardiac-specific promoter, which is alpha myosin heavy chain. Level number 2, the CRE is not a simple CRE. It is a CRE with the mutated estrogen receptor. Therefore, it realized that it doesn't work unless I add tamoxifen. And the third condition is that I'm not using a knockout, I'm using a LOX-P on the other side. So this is the third level of control. So one of your colleagues asked me how you can control when the CRE will work and where you can control the CRE can work and at what stage you can control the CRE can work. So in this slide here, you can control everything. You can control which cells because it's promoter specific to cardiomyocytes. You can control which gene because you have the locks beside which age or which time, because you have a conditional knockout, not a general knockout. And when you want to do it, when you cross these together and you add tamoxifen. If you don't have tamoxifen, these mice will not be having any issues, no knockouts. So the inducible conditional gene targeting, it's controlled by cardiac-specific mutagenesis that can be achieved by specific expression of the CRE-ERT2 transgene or in a knock-in mouse. So here it's you knocked in the CRE-ERT2 into the cardiac-specific promoter, which is the alpha-amylin heavy chain. And then the specificity of the CRE-ERT2 depends on if you add tamoxifen or not. Otherwise, the CRE is not active. So in the absence of tamoxifen, CRE-ERT2 is not retained in the cytoplasm. Only after binding by tamoxifen, then the CRE-ERT2 will go and bind to the LOXP sites. In this condition, it's exon number 2 for whatever gene. And then it will delete that gene. So this is a knock-in animal. This is a conditional knockout animal. And together, they generate a tissue-specific inducible knockout. Inducible conditional gene targeting. Okay? This is everything I will be describing for you today, and I will stop to take any questions. Clear? So now, in your assignment number 3, I have sent you one of my papers that I used probably 21 different transgenic animals. I selected some of those where we have genes that are fluxed with LOXP site, and we have certain cardiac specific and fibroblast specific CRE lines. So all you need to do is just read through this lecture and remember what is the type of each of those genes. It's a tissue specific, it's a conditional knockout, it's a general body knockout, it's a whole body knockout. It is the, like the questions are, I don't remember all the questions I put in the assignment. You have 1 month almost to do that. I created a discussion for general questions. I really suggest and recommend strongly to use this discussion to put your questions there, and I will be answering them so all your colleagues will see. And I wish you good luck with that. And with this, I think we will stop discussing any more about DNA. We will switch in the next lecture for isolation of RNA and techniques for studying the RNA. And the lecture after that, we will start discussing the proteins. Any questions, any comments, any concerns? Anybody?
Professor, sorry,
Yes.
just to make sure that I'm understanding this right, the difference between general and conditional knockout, it's not that like you're still generating like first generation and second generation in conditional, right? But it just has the CRE gene.
Yeah, the difference is the difference is in the targeting vector. So the targeting vector in that situation, you're simply taking out an exon and put in a neocassette permanently, while in the conditional knockout, you are introducing the LOXP sites around that exon.
Right.
You still have to do exact same procedure from A to Z. You
So
have
like
to,
same thing, like you're still looking for the homozygote, but
yes,
just that the gene is inducible.
yes. So
Okay.
the deletion is inducible, you want to say.
Okay. Thank you.
Yeah. Okay. So I don't think anybody wants to discuss anything related to the classes, the course in general. You want to stay on your own? You don't want your colleagues to hear what you say? Please, if you want, I will stay a few minutes. Okay, I'll stop sharing. I'll stop recording.